Hello, Biology 230. This is Dr. Chastity Bradford, an assistant professor here at Tuskegee University in the Department of Biology. And I will cover Chapter 6.4a as I continue to be your tour guide as we tour the cell. 6.4a covers the endoplasmic reticulum. It covers the smooth and rough ER, as well as vesicles. The endomembrane system regulates protein traffic and it performs metabolic functions in the cell. What is the endomembrane system? You've heard about parts of or components of this system in previous lectures in Chapter 6. But let's talk about it in its entirety. So there are 6 components within the endomembrane system. You must know that the endomembrane system is composed of a nuclear envelope. It's composed of the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and plasma membrane. So all of these are components of the endomembrane system. You will be tested on this, and you will have to know these components and their functions. We've already discussed the function of the nuclear envelope. We've already discussed the function of the plasma membrane. We will go into more depth with the plasma membrane. But in this lecture, we will review the endoplasmic reticulum. And it's good to know that these components are either continuous or they're connected. And they're often connected by vesicles. The ER is often seen as a biosynthetic factory. You think of factories, there's a systematic way that things are made. And so our body has a biosynthetic factory. And the ER, or the endoplasmic reticulum, not the emergency room, it accounts for more than half of the total membrane in many eukaryotic cells. So the ER, or endoplasmic reticulum, its membrane is continuous with the nuclear envelope. And I'll show you an image in the next slide. And there are 2 distinct regions of the ER. There's smooth, and there's also rough ER. The ER, it winds itself through the cytoplasm. So if you look here at the depiction of an animal cell, you'll see that there is smooth endoplasmic reticulum here within the cytoplasm. And you see that there's rough endoplasmic reticulum here within the cytoplasm. And so it winds itself through the cytoplasm. Also in a plant cell, there's rough ER, and there's smooth ER, the ER with outbound ribosomes. And it winds itself through the cytoplasm. This is the cytoplasm. All of this in yellow would be the cytoplasm. It winds itself through the cytoplasm within this plant cell and within the animal cell. What does this do? The fact that it's winding itself and it's compact in this way, it is providing increased surface area. Recall from the previous lecture the importance of surface area to volume ratio, okay? And how in eukaryotic cells, how just this compartmentalization increases surface area. The endoplasmic reticulum is also continuous with the nuclear envelope. So this is smooth endoplasmic reticulum, and this is rough endoplasmic reticulum. The rough has bound ribosomes. Ribosomes do what? Make proteins. So notice that this is the nuclear envelope here in purple, and that the ER, the endoplasmic reticulum, is continuous with it. So the nuclear envelope ends here, the ER begins here, so they're continuous. increasing surface area. What's the function of the ER? Let's look at the function of the smooth ER first. This is important. You must know and understand that the smooth endoplasmic reticulum is responsible for synthesizing lipids, metabolizing carbohydrates, and detoxifying. It also stores calcium. Let's look at some examples of lipids. Sex hormones are examples of lipids. Testosterone. And so if you were to look at organs that have sex hormones, such as the testes for the male or the ovaries for the female, one would expect to find more smooth endoplasmic reticulum, because this is where synthesis of lipids takes place. This is how you have to apply the knowledge. What about detoxification? Often when people are on drugs and they go through a detox program, one of the examples of detox in one of the ways in which a detoxification will work is that the drugs that are given to the patients, the detox drugs, if you will, they add OH groups. What are the OH groups? You should know this functional group by now. They add hydroxyl groups to the drug, okay? And that detoxes or removes the drugs from this person's system, okay? And this takes place in smooth ER. Let's look at the structure of both smooth and rough ER, up close and personal. So this is smooth ER. Look how continuous it is. Look at this network. This network of tubules is called cisternae. Network of tubules, okay? Again, this is smooth ER and this rough ER with the bound ribosomes. Ribosomes are attached to the cytosolic side of the rough ER in this example. And the lumen of the ER, of this rough ER, you see that? The lumen is just representing an open area, okay? This is a lumen. So this would be a lumen here as well in the smooth ER. So it's important that you begin to learn these terms because they apply to a number of organs and different structures. So what are the functions of rough endoplasmic reticulum? The rough ER, which has bound ribosomes, they secrete glycoproteins. Glycoproteins are proteins that are covalently bonded to carbohydrates. Glycoproteins, proteins covalently bonded to carbohydrates. Covalent bonds. Reminds me of a previous chapter. The rough ER distributes transport vesicles. It distributes proteins that are surrounded by membranes. The rough ER is a membrane factory for the cell. And these vesicles are like the trucks of the endomembrane system. They function to store, transport, or digest cellular products and waste. The rough ER, we talked about how it secretes glycoproteins. And if you recall from one of the previous video lectures in this chapter, we talked about the plasma membrane. And I showed you this picture, and we went through the different types of proteins that were within this membrane, but also highlighted glycoproteins. So these glycoproteins are secreted by the rough ER. And look where they end up in this plasma membrane. The rough ER is also responsible for distributing transport vesicles. So if this is ER, and I said rough ER, and you don't see any what? Buds here. You don't see any ribosomes here. So if this is rough ER, what I want you to get from this, let's say they're on the other side, okay? The ribosomes are on the other side. So this rough ER distributes transport vesicles. So you see these vesicles budding off of this rough ER? It's amazing how this happens within our body. These are transitional endoplasmic reticulum, transitional, moving things from one place to the other. This is the lumen of the rough ER in this example. The lumen, just these openings, okay? So these transport vesicles are budding off. And guess where they're going? They're going to the Golgi. So what else do we know? What else takes place? Proteins are modified in the lumen. And one example of a modification is that a lipid could be added to a protein. Okay? So this vesicle is in transit to the Golgi. As a protein is being made, modified in the lumen, one of the modifications is that a lipid could be added to it.